So this is just a summary of the current evidence for fecal microbiota transplant as a treatment for depression. So the microbiota-gut-brain-axis is something that we've known existed for quite a long time, but we haven't really fully understood its function and how it works. So this essentially allows a bidirectional crosstalk between the brain and the gut microbiota. And it's thought to play a key role in various body functions, including mood, cognition, behaviour, metabolism, biological processes, and overall homeostasis. The manipulation of the gut microbiome through fecal microbiota transplant is a new and possibly very promising treatment for major depressive disorder, for which we have quite a limited range of treatments. So studies have shown quite consistently that there is very significant gut dysbiosis in depressed patients compared to healthy cohorts. So the most notable findings are overgrowth of pro-inflammatory microbiota, reduction in anti-inflammatory species, and then overall less stability. FMT essentially allows healthy microbiota to be introduced into the GIT of the individual suffering from depression, and this leads to restoration of the eubiosis. In healthy states, the gut microbiome is usually quite stable, and there's a constant relationship, two-directional relationship with the host. And studies have shown this kind of dysbiotic picture in treatment-resistant patients compared to treatment-responsive patients. So there have been a lot of individual studies, case reports, laboratory studies, and randomized controlled trials. And this is just a summary of some of the evidence that we have so far. So in depressed patients, there are specific inflammatory species that are more common, including ruminococci. And then the reduction in anti-inflammatory bacteria, specifically Saturella. And then, interestingly, addition of these pro-inflammatory species to duloxetine treatment in depressed mice was able to reduce the effects of the duloxetine overall. So this makes us question whether these microorganisms could actually play a part in the disease process itself, particularly in treatment-resistant cases. And there's this question that we haven't been able to answer yet. Is the dysbiosis a cause or a consequence of the depressive state? This diagram just very briefly summarizes the microbiota gut-brain and the communication channels that have been identified to play a role. So these include the immune system, vagus nerve, neuroendocrine system, circulatory system, and the enteric nervous system. It's likely that all of these different systems work in combination to result in the bidirectional crosstalk, and quite likely that there are other avenues of communication that we're not entirely aware of yet. Gut microbiome is thought to play a role in one's physiological response to stress, and as we all know, stress is directly associated with the development of depression, and stress is also known to directly affect the gut microbiome composition. Fecal microbiota transplant is for diseases associated with dysbiosis, for example, major depressive disorder. So the aim here is to manipulate the gut microbiome within the intestinal microenvironment. And this is by inoculating fecal microbiota from a healthy donor into the recipient's GI system. And the FMT can be performed in a variety of different ways. The current evidence from rodent studies, from human studies, and from case reports has all been very, very promising. And one of the notable studies showed that an FMT was performed on 49 patients with recurrent C. difficile infections. Now, the C. difficile diarrhea improved in these patients when they were given FMTs from healthy, and anxiety scores significantly improved, and these effects were maintained for up to 6 months. The reverse has also been shown to be true. So inoculation of microbiota from depressed individuals were able to cause depressive symptoms in healthy individuals. So, in conclusion, the gut microbiome plays an integral role in human health in many different ways. Current research supports the ability of this to be able to modify the human microenvironment and overall human health and homeostasis. Studies have shown specific taxa which are commonly associated with mental health disorders, and the specific genera that are found in lower levels in these individuals. The FMT can transfer the phenotype into the recipient, although we need to be careful because it can transfer both positive and negative phenotypes. The gut microbiome can be manipulated in a number of ways, but we need further research to determine the exact profiles associated with depression and the long-term effects and potential risks of this treatment. Thank you.